Yeah, so it's great to have a chance to present our lab's work. I'm from the University of Washington, but I actually grew up in Chicago, so it's a nice homecoming to be able to present here. I'm going to be talking about using long-read approaches to better understand genomic and epigenomic contributions to cancer, and I'm going to be speaking about retinoblastoma, which is a rare form of cancer, but it's the most common pediatric intraocular malignancy we see as retina specialists. This work has been subject of a patent, otherwise I have no other disclosures. Molecular diagnostics has gone through a revolution. I remember when I was a grad student, I cut my teeth doing short read sequencing, and that was all that was available at the time. But things have changed, and what we have started to learn is that there can be a lot embedded in this genetic and epigenetic information that we have been seemingly glossing over time. More importantly, we've started to understand, and I think other speakers will talk about this too, the understanding of genetic and epigenetic contributions to not only cancer, but risk factors as biomarkers, understanding which cancer may be more progressive, more aggressive than the other ones, and actually using that to guide surveillance and treatment. It also provides us with a way to understand individual and familial recurrence risks. So parent of origin is very important when we're trying to initiate cascade testing. And if we can do that from the very beginning, it kind of eliminates the risk of having to go through every family member to figure out who may have the cancer variant and who may not. The downside of current clinical testing systems is that it can be very complex. And sometimes we get incomplete familial risks, oftentimes because we cannot do full familial segregation, especially in cases of de novo variation. So I won't belabor the standard clinical testing that currently occurs, but it can be onerous in the fact that oftentimes these arrays that we design or that we are sending off are built for very specific genes. And to update that can be onerous, and it cannot be something that we can necessarily do. We cannot tell a company to change their cancer panel. But what Dr. Atwell touched on and what I'll talk about today is that with ONT technology, you can change your cancer panel in a minute by changing your bed file. And you've decided that there's a new gene that you want to test or take out a gene. It's as simple as a change in one file and complete change of your entire panel without having to do anything with your input sample. The biggest thing that we're able to do with the ability to sequence longer stretches of DNA is uncover things that we did not know potentially existed. I always like to think about doing a puzzle. So when we're putting these reads back together, we're essentially stitching back a puzzle. And with short reads, you have a 5,000 piece puzzle. And for anyone who's done a puzzle before, a 5,000 piece puzzle is no joke. You're going to lose pieces. A lot of pieces look the same. You have ambiguity, and it's really hard to figure out what goes where. Long read essentially takes that 5,000 piece puzzle and makes it a 5 or 10 piece puzzle. You have overlapping pieces. It's very easy to do. My toddler is really going to be able to do this. And that's the ability for us to do, is put things together that we could not before. And this opens up for us to understand things like non-unique sequences, like pseudogenes, repeat expansions, structural variants. And what I'm going to really touch on today is phasing. This is a fundamental problem in medical genetics, is understanding haplotypes, is understanding who contributed what and how could that be relevant, especially in autosomal recessive diseases. The standard way we do it is usually familial segregation or population-based genetics. So at the top here, you're seeing that you're getting multiple family members. You're trying to see who has what in common so you can phase a particular variant as if they're either in cis or trans. But if you're able to do this with long-read technology, you're able to get a fully phased understanding of the variant profile in an individual. So, the setup for us is very simple. We both use saliva and blood, and this could be however you want to do it. We use magnetic base extraction, but for high molecular weights, we'll use glass bead extractions. And ultimately, with your high molecular weight DNA, you're able to ligate your adapters, and that's essentially all you need to do. You're able to preserve the native structure, so you're not doing any kind of amplification for your DNA or RNA in this case. And I'm going to walk you through, if anyone wants to stay, I would recommend doing one of these loadings because it is very simple to load a flow cell. But we run the MinIONs on our workstation. We have a very powerful GPU that allows us to do adaptive sampling. But the basis of all this is housed in this ASIC that has a sensor chip and an array of proteins that really are going to guide your RNA through. Once you put your flow cell buffer and you load your protein onto the load your DNA onto the protein microarray, you're ready to sequence. So you turn your computer on and you see what this screen looks like. And if you zoom in on one of those boxes, what you're really looking at is a single protein nanopore or a cluster of 4. And as Dr. Atwell pointed out, what you're really doing is you're allowing these DNA molecules to go through. And because they're negatively charged, you're going to generate a current. So as each one goes through, you're going to generate a picoamp current. Now, this is dramatic. We're not looking at picoamp currents, but each different base is going to give you a unique signature. And what's important here is that there's going to be a cytosine, and a methylcytosine is going to give you a different signature. And when you sum all those up, over a few hundred base pairs, we have deep learning tools to actually predict what that sequence is going to be with very high accuracy. And the beauty is you can look at 5MC, you can 5HMC, 6MA, many different modifications, because we have used deep learning tools to teach them what these different squiggles really look like. And with this in hand, you can do things like adaptive sampling. So, when I started my lab, I really was interested in looking at specific genes. We had a hypothesis of what genes were contributing to retinal diseases and retinoblastoma, and we really wanted to look at those genes. So for us, at the top here, if you do a whole genome run, you're not getting the information you need. You really want to get high coverage of your genes of interest. For example, in this area, we're looking at 5 specific genes, but in each gene, we're getting pretty low level coverage. So if you have a variant, you're not going to be able to phase it. You may not even cover it if you're unlucky. What adaptive sampling does with an input bet file is it focuses your sequencing depth to the regions of interest. So in this case, we now have 4 different targeted regions. We do get coverage in between, but these are short reads. So these are 200 to 400 base pairs based on how quickly you're able to do adaptive sampling on your computing workstation. So it's kicking out all these reads in between, and it's really reading these giant pieces of DNA that fit into your profile. So what this means, as Dr. Atwell pointed out, this is for a different assay of 370 genes, you get really robust coverage. So we're getting 25x coverage on a single min-ion flow cell compared to the background coverage. And the biggest thing it translates to is phasing. So the phase breadth is near 100% when we do adaptive sampling. Well, that number is between 70 and 80% when we are not able to do that, or we just look at the whole genome channels. And what this relates to is being able to do very precise haplotype analysis. So in this case, we have a large structural variant in the second haplotype and a single nucleotide variant that we can very accurately phase. And we can see that not only is this knocking out the very beginning of the gene, but it's going upstream of exon 1. And the other thing that we can now look at is methylation signal. So we can start to look at epigenetic signatures underlying certain diseases. So in this case, we have the benefit of actually having retinal data to look at cis-regulatory elements from ATAC-seq and methylation profiles and acylations of different histone modifiers. And we can see that these methylation profiles that we're getting from blood can oftentimes reapproximate what we see in the tissue themselves. So, that's a summary of what our lab does on the non-cancer front, but I'd like to focus the rest of this talk on retinoblastoma. For those of you unaware, retinoblastoma is a very devastating cancer in the pediatric population. The worst burden is in countries that are underserved. So even though the survival rate can be in the 90%, 97% in developed countries like the US, that number is much lower in sub-Saharan Africa, where retinoblastoma is a true epidemic. Now, retinoblastoma is really divided into 2 classes. The first class is somatic mutations, so non-heritable. So these patients are born without a germline mutation, and they get 2 hits in somatic changes in the retina. And if we catch these, there's no risk of extraocular cancer. But 40% of the time, there's a germline mutation. And that second hit not only can dictate their risk of extraocular disease, but also how they will respond and their treatment regimen that we have to do as pediatric retina specialists. Now, of those 40%, 90% of those are de novo. So only 10% are familial. So if you look at a mom or a dad and you'll say, okay, mom had retinoblastoma, so the child got it, right? Now, oftentimes that may not matter. What does it matter if it's de novo or familial? Where it turns out that parent of origin actually plays a huge role in disease prognostication in retinoblastoma. So there's a first few papers in the 2016s and the most recent paper in 2024 that shows there's a variation in disease presentation, treatment outcome, and overall prognosis of the eye. We don't know much about secondary cancers yet, depending on who the disease came from. And they found that paternal inheritance was associated with worse disease burden and risk of enucleation or loss of eye compared to those who inherited the same mutation maternally. Now, as I mentioned, this is a very important prognostic information, but it's only apparent or available to 10% of the population that have heritable cancer, because the 90% are de novo. So even if you go back and test mom and dad, unless they happen to have a hypomorphic variant, you're not going to find anything, right? So this is what retinoblastoma looks like in the eye. This is a very well-treated girl, about 2 years old, that we saw. She has tumors that have responded really well. And as we talked about, we can do targeted sequencing. So we targeted the RB1 gene, and we can get really great genomic coverage. So that's the top panel up there in the yellow and purple. So we can differentiate between haplotype 1 and 2. Not only that, we can then look at the methylome. So we can look at haplotype 1 and 2. And we can zoom in. There's dash boxes that are going to show you the promoter region going up to where the mutation is, all the way to the right of the screen that is pointed out, the blue 542 stop. And what you may notice is that right in the middle, so the promoter is uniformly blue, which means it's unmethylated, which is often the case of these genes when we see it in blood. But there's an imprinted signal in RB1, and that imprint is able to tell us parent of origin. So unmethylated tells us that it's a paternal origin versus methylated, red, telling it's maternal. And this patient happens to have her variant on the bottom red allele of the imprint region, which tells us it's maternal. And we knew this was maternal. And actually, this was not something that we discovered. This was discovered in 2009. But unfortunately, got lost in the literature because it was very hard to do whole genome bisulfite sequencing and link those reads to variants that were actually causing cancer. So back then, they only analyzed 12 of the CPG sites in that intron. They found this methylation pattern, and it was kind of lost to the literature, unfortunately. What we're able to now do is show that there are not only, there's a parent of origin that we can determine, but there is an effect. So in this patient, for example, we just showed you that she has known maternal inheritance. So she has her variant that segregates onto the methylated region of imprint, the imprinted region in intron 2. If you look at mom, it's completely flipped. So she has the same variant, but it is segregating on the unmethylated region of intron 2. And why is the case? That's because she got it from her dad. And it turns out that even though these two patients have the same variant, they have very different forms of disease. The daughter, who we have been treating for some time, is very stable, has not had any recurrence of disease. The mother had very aggressive disease as a child. So what this tells us that the same variant, we have been always thinking in retinoblastoma, potentially other cancers, that you have the same variant, this is going to be your progression. But at least we're learning from this cancer that your variant can be the same, but you could have a very different form of cancer based on your parent of origin. So we've now validated this in many known cases. So 8 known cases, we made sure that can we identify the parent of origin, and does it actually relate to what the familial data tells us? But what's most important is now we can assign parent of origin in de novo cases. So patients that we did not know family history of, and we can start to say you have a inheritance on the unmethylated or methylated allele of intron 2, denoting maternal or paternal inheritance. And ultimately, what this allows us to do is not only can we get a very rapid diagnosis. So for us, diagnostics is very important because when we see a patient, not only do we have to identify the variant, we have to do that very quickly because it may dictate their treatment profile, how often we're bringing them in for exam under anesthesia. And because we can do adaptive sampling, within 12 hours of sequencing, we can pull our data off. And not only can we get full haplotype analysis, we can get methylation signaling, and we can not only diagnose them with the variant, but we can also give them a parent of origin diagnosis as well. And again, this is very important for a variety of cases. For clinical cases like mosaicism, this could take months for a clinical system to figure out, which we're able to rapidly find out because of the enhanced depth of coverage. And it can phase distant regions. So this is a patient that had a very complex form of retinoblastoma where they also had multiple other comorbid conditions. And we were able to sequence all of chromosome 13 and identify a very precise breakpoint that happened between a translocation between chromosome X and chromosome 13. And interestingly enough, we think that this led to disease in this patient, not because of a issue in retinoblastoma, but because of silencing. And we can start to see some aberrant promoter methylation in this patient as well. And ultimately, the big punchline of all this is that parent of origin provides prognostic information. When we took all this data of familial, de novo, everything that we have diagnosed, it turns out there is a huge difference in survival and risk of chemotherapy failure between maternal and paternal samples. It's a significant association that we see, and it really is going to dictate how we start treating these patients. And I just want to end with the last minute to talk about that. This is a very unique case. Not every cancer gene is going to be imprinted, but I don't think you need every cancer gene to be imprinted. All you need to do is find something that is imprinted nearby. This is an example of using RB. So BRCA2 is a very well-known cancer gene. It is not imprinted, but it is close enough to RB1 that we can phase that entire region with a single run to show that not only can we identify potentially variants in BRCA2, but we can assign paternal or maternal origin of that, which can dictate cascade testing. So I just want to end by saying that, I think what this approach really has allowed us in our lab is to give it a more complete picture of disease variants, genomic modifications, all in one run. It leads to better informed decision for our teams. We have been using an IRB to return results to families and now it's dictating our treatment regimens in the hospital. And I think the ability to assign a genomic variant with a local or distant epigenetic signal is going to better allow us to understand parent of origin in cancers and many other diseases that we just did not have access to. And I just want to say, if anyone does work on retinoblastoma, we have a clinical trial trying to understand the genotype of extraocular and ocular disease in a large patient cohort. And with that, I just want to end. If anyone's interested, the code on the bottom left there is a link to our paper that we just published on retinoblastoma and parent of origin. And I just want to thank my team. I get to talk about all this stuff, but they're the ones who are doing all the work and all of our funding, and especially the Gerber Foundation, who's really funded the lion's share of this work.